Let us start with the first kingdom, Monera. The common features of Monera Kingdom are number one, and this you have to keep in mind always that whenever we talk about Monera, they are always prokaryotes. Pro means primitive, and karyon terms is used for nucleus. So all the organisms which are very primitive in their nature and which have a very primitive nucleus are known as prokaryotes. And all the monerans are unicellular organisms. So these are the two basic character of Monera kingdom, prokaryote and unicellular. Another thing is that their DNA, their genetic material, they have a DNA. DNA is circular, naked. Naked means it is not covered in a nucleus, it is not enclosed by any nuclear envelope. They have one single double-stranded circular DNA. Ribosomes are of 70S type. I have explained about this in the last video. Sap vacuoles do not occur. We have studied in plants that plants have vacuole. They have a different type of vacuoles. In Monera kingdom, they may have gas vacuoles, but not the other type of vacuoles. Monera organisms of Monera kingdom are mostly saprophytic or parasitic. They have an absorptive type of nutrition, but some groups of Monera are photosynthetic or chemosynthetic also. Children, here you have studied a new term, you have got a new term, chemosynthetic. I'll explain about this again. The organisms are non-motile, or they may have motility or movement, and for movement they have flagella. Flagella is made up of a protein, thread-like protein called as flagellum. Moneran cells are microscopic. The size of these cells are in microns. 1 micron is equal to 10 to the power minus 6 meter. So they are 1,000th part of 1 millimeter. The cell wall of Monera kingdom is a very complex structure and it is made up of peptidoglycan. The production primarily asexual by binary fission. Examples are archaebacteria, eubacteria, actinomycetes, mycoplasma, and cyanobacteria. Now, the shape of bacteria. Bacteria are found in 4 different shapes. Spherical, known as coccus, rod-like, bacillus, comma-shaped, vibrio, and spiral-shaped, spirulinum. Children, this is a structure of a common Moneran cell. As you can see, it has a 3-layered covering. The outermost is capsule or slimy layer. The second one, cell wall. And the last is plasma membrane. The innermost is plasma membrane. And inside the plasma membrane, the liquid filled is known as cytoplasm. Here it has not a proper well-defined chromosome, but it has DNA and it has very few proteins along with the DNA. This region is known as nucleoid region. They have flagella, food granules, ribosomes, villi, cytoplasm, and one more thing, here it is written plasmid. It is again a different DNA other than nucleoid. So we'll discuss about all these structures. So let us start with cell wall. Cell wall is a hard covering. It is made up of polypeptide and polysaccharide. Now children, in living organisms, there are thousands of the big molecule present. Now these molecules are grouped in different categories, different biomolecules like carbohydrate, protein, lipid, DNA, RNA, etc. So polysaccharides are a type of carbohydrate, whereas polypeptide is a type of protein. It is a part of protein. Now, as I say, we have a different around thousands type of polysaccharides also present in our body, even in all the living organisms, and different type of polypeptide. In bacteria, these are peptidoglycans or murine or mucopeptide. They are the different names of polysaccharide and polypeptide. Now, when you are studying different books, you are following any other book, you must be getting different names. But as it is not a single molecule, it's a different type of molecules. And as I said, in bacterial cell wall, different type of polysaccharides and polypeptides are present. Even let me add here that in bacteria, based on their cell wall, they are categorized into two types: gram positive bacteria and gram negative bacteria. Gram positive bacteria are those bacteria whose cell wall is thick, and gram negative bacteria has thin cell wall. So we'll study about gram positive and gram negative bacteria in detail afterward. Along with the cell wall, bacteria also have outer polysaccharide layer of slimy layer. Now this slimy layer makes the bacteria wet, and it protects the cell from desiccating. Bacteria needs oxygen from its surface. It requires oxygen or food which is dissolved in its outer membrane. So slimy layer is the layer which always keeps it wet so that bacteria can absorb oxygen from outside. Now when this bacteria are present in air or in dry conditions, this slimy layer becomes very hard and forms a capsule. When it forms capsule, bacteria can remain as such for several months to year even without doing any metabolism. The innermost layer is plasma membrane. Children, plasma membrane is a structure which is found from bacteria till animal kingdom everywhere, and it has the same structure. In bacteria, it is made up of lipid and protein, and same way it is found in plants and animals also. So plasma membrane is made up of lipid and protein structure, and it is selectively permeable. Selectively permeable means it is semi-permeable. It allows only selected particles to cross through it. This plasma membrane is enfolded inside and is known as mesosomes. अंदर की तरफ में प्लाज्मा मेम्ब्रेन बहुत ही मुड़ी चुड़ी सी एक स्ट्रक्चर बनाती है जिसे कहते हैं mesosomes. This mesosomes increases the surface area. Why surface area is required to be increased? At the time of binary fission, जब बैक्टीरिया को अपना साइज बढ़ाना होता है और उसको दो पार्ट में डिवाइड होना होता है, उस समय में यही मीजोसोम्स सरफेस एरिया बढ़ा करके और बैक्टीरिया के साइज को इंक्रीज करते हैं. Now, it has been found that mesosomes also contain different enzymes, and so it helps in respiration of bacteria also. Bacteria have different type of food reserves. It has glycogen or lipid stored in the form of different globules. Nucleoid, I have explained, it has one circular double-stranded. Double-stranded means two threads. DNA is made up of two threads. So this is a genetic material, and this is known as nucleoid. Flagellum. Now, flagellum is a singular term. Flagella is plural term. This flagellum or flagella is found in those bacteria which are motile. And you know that coccus bacteria are non-motile. So flagellum, it's made up of flagellin protein, and it has three parts: basal body, hook, and filament. Flagellum arises from the outer structure of plasma membrane, and its tip is known as basal body. The part of flagellum which is present in the cell wall and slimy layer, is known as hook. And it has a long filament which moves like row of a boat and help in movement of bacteria. Along with the flagellum, bacteria also have tiny uh, hair-like appendages, as you have seen in the diagram, these small structures, spiny structures, which are known as villi or fimbriae. The difference in flagellum and villi is flagella number is less and their size is longer than villi or fimbriae. Now, these are the two different structures. When this hair-like structure is used to adhere to the surface, it is known as fimbriae. But when these structures are used to attach with another type of bacteria, it's known as villi or sex villi. Now, you must be thinking that why have I written here male bacteria to female bacteria? Children, in the previous slide, when I explained that sometimes there is a slight exchange of genetic material between two bacteria. So that means two bacteria, they attach to it, they come closer to each other, get attached, and DNA of small part of DNA from one bacteria is transferred to another bacteria. So when the bacteria who gives the genetic material is considered as positive or female bacteria. Another bacteria which gets the DNA is known as negative bacteria or male bacteria. Ribosomes, bacteria has 70S type of ribosomes, which is made up of RNA and protein. In ribosomes, there are two units, 40S and 60S. So here you have to understand that this is not a simple addition. It's based on the sedimentation. Sedimentation means when these ribosomes are extracted from the organism and it is rotated very fast, they get settled at different points. And these points are termed as 10S, 20S, 30S, 40S, and so on. So the position where they get sedimented is the term given to them. So when both the units are together, it settles at 70S. But when these two units are separated, the smaller one settles at 40S and larger one at 60S. Function of ribosome is the same as you studied in class 9. It is protein synthesis.